The overall goal of this procedure is to obtain whole cell voltage clamp recordings from a single photoreceptor cell to measure light-induced currents arising from single photon absorption and from repeated responses to intense light.
This method can help address key topics of the sensory transduction field, such as visual transduction, the biophysical and ionic mechanisms that underlie phototransduction, and the properties of TRP channels.
The advantages of using this technique includes full control of the membrane voltage, an extremely high signal-to-noise ratio, and the ability to introduce specific indicators for optical measurements.
Generally, individuals new to this method will struggle with the minute size of the perforation and the need to perform the dissection quickly due to its high metabolic vulnerability.
For this procedure, raise flies at a density of about 20 per vial. At the time of the experiment, have a few vials with pupae that are about to be closed and have been under constant darkness for at least 24 hours. 2 hours prior to collecting adults, remove any eclosed adults. Always handle the dark-adjusted flies in tissues under red light illumination. While waiting for the freshly eclosed flies, prepare the recording chamber. Onto the bottom of the chamber, fix a cover slip using melted paraffin or high vacuum silicone grease to complete the bath. Then mount the bath to the stage of the inverted microscope. Into the bath, place the perfusion system tube, the suction system tube, and the ground wire for the